Hi guys, Miss Francis here. Today I'd like to discuss linked genes. So what does it mean if something's linked? It means that they're connected. Well, genes can also be linked. What does it mean when genes are linked? It means that they're located on the same chromosome and they tend to be inherited together. Think about someone who has red hair. They usually have freckles. That probably means that the gene that codes for red hair and the gene that codes for freckles are located on the same chromosome. And how do we know about these linked genes? Because results of crosses with linked genes deviate from the results of those expected according to Mendel's law of independent assortment. So let's take an example. So Mendel investigated two different traits. For example, well, he investigated more than two different traits, but let's take two traits. Let's look at pod color and flower color. And Mendel did dihybrid crosses where he took a parent that was heterozygous for both traits, and then a parent that was recessive for both traits, where this would look like the genotype for one parent, and then this would look like the genotype for the other parent. And when Mendel performed this dihybrid cross, the ratio that he found was a 1:1 to 1:1 ratio, where each one of the phenotypes was present in a quarter of the offspring. So the phenotypes came to a 1:1 to 1:1 ratio. Well, Morgan, with his fruit flies, did experiments that were following Mendel's pattern of inheritance. So he decided that he too was going to investigate two different traits in fruit flies and see if they still follow Mendel's patterns of inheritance. So Morgan decided to investigate body color and wing size, where he looked at the wild type for body color and wing size and the mutant type for body color and wing size. So for body color, wild type is gray, mutant is black. For wing size, wild type is normal and mutant type is vestigial. So what Morgan did was he did a test cross. But before we even get to the test cross, let's note that what happened was Morgan did not find a 1:1 to 1:1 ratio. Instead, he observed a large number of the offspring that were wild type and double mutant, the phenotypes that correspond to those parents, but he also observed a small amount of wild type mutant gray vestigial and mutant wild type black normal flies among the offspring. So let's take a look at what Morgan actually did. Looked at the two different traits, body color and wing shape, and he looked at the wild type and the mutant for each one of those traits. Now the pictures kind of use this notations. I changed it to this just for simplicity's sake. So follow along with me and let's see what Morgan did. So Morgan took his parental generation and he took a true breeding female with a true breeding male. That means that this female was dominant for both traits and this male was recessive for both traits. And when he crossed those two flies, he got what he expected. All heterozygous offspring. So he got, in this case, he took the female that was heterozygous and did a test cross with a male that was a double mutant, meaning that it was recessive for both traits. And what he should have gotten was this 1:1 to 1:1 ratio. That would be following Mendel's pattern of inheritance that he created his law of independent assortment from, where one trait did not impact the inheritance of another trait. But what Morgan observed instead, so here's what he actually observed. He observed a large number of the parental phenotypes, but he also observed a small number of what we call these recombinant phenotypes, where the offspring were mutant for one trait and then wild type for the other trait. So let's take a look in words at what Morgan did. So Morgan's experiment that discovered that genes can be linked involved a parental generation where one parent was homozygous dominant and the other parent was recessive for both traits. And in the F1 generation, Morgan got the results that he expected. According to Mendel's laws, I got 100% heterozygous offspring for both traits. Now, as Morgan bred his F1 generation, what he expected to get was a 1:1:1 ratio. So he did this test cross where he took a female from the F1. That female from the F1 was heterozygous dominant for both traits. And the male, the one that acted as the test cross, was recessive for both of the traits. And what he ended up with was not the expected 1:1:1 ratio. The expected is a quarter of each one of those phenotypes. But instead, what Morgan saw, as we said earlier, was a large number of wild type and double mutant flies, which were the phenotypes that corresponded to the parents. And then he observed a small number of wild type mutant gray vestigial and mutant wild type black normal among the offspring. So what Morgan reasoned was that, well, body color and wing shape must be inherited together most of the time because their genes are on the same chromosome. And then this is what it would look like. So if I look at the mom's chromosomes, right? Here I have the dominant allele. Here I have the recessive allele. And if I look at Dad only had the recessive allele for both traits, because that's what a test cross is, where you cross something with something else that you know is recessive for both traits. And what Morgan observed in most of the offspring was he got this parental phenotype. Oh, geez. Oh, God. Where he ended up with the parental genotype, right? But then there were some that did not look like this, the unexpected phenotypes. Now, if these genes were completely linked, we should see a 1:1 to 0:0 ratio. There would be no offspring that don't look like the parental phenotypes, but that's not what Morgan saw. So he said there must be some sort of recombination or crossing over, some mechanism by which these alleles are being reshuffled. Where in Morgan's case, he ended up with 17% of the flies being this recombinant type. So what Morgan proposed was that there's a mechanism where segments are exchanged occasionally between homologous chromosomes. Crossing over occurs, and we call these recombinant chromosomes. And this mechanism we now know is called crossing over, which actually occurs during prophase 1. So what would this actually look like? Well, let's take a look at the female's duplicated chromosomes that happen before meiosis 1, right? So here I've got my female chromosomes, where on each chromosome I have both allele, and then I've got my homologous pairs of that chromosome. Meiosis 1 occurs, and it's during meiosis 1 that prophase 1 occurs. And then during prophase 1, what happens is these non-sister chromatids between homologous pairs exchange alleles. I like to say they play footsie with each other. So we end up with 4 different potential, in this case, eggs or ova. So here in meiosis 2, when my homologous chromosomes separate, I could have my first potential egg. Then I could get my second potential egg. Here would be my third potential egg. And here would be the chromosome that would be found in my fourth potential egg, where these last 2 eggs are recombinant. They contain alleles from the homologous chromosome. So taking this to Morgan's experiment, let's see what the chromosomes would look like. So here I've got my test cross that involves the parents, right? So I've got my female that is heterozygous for both traits, and she can create these 4 potential eggs. Now the male is recessive for both traits. So the male can only create this one type of sperm. Now when these eggs get fertilized by this sperm, I end up with these 4 potential offspring, where most of them look like the parental type. Most of them have the same phenotype as the parents. However, there is a small portion that are known as recombinant. They have some of the mom's allele, but they have part of the dad, which is his allele. Now, when I do the math for this, I end up with 17% of my offspring being recombinant. We're going to use that percentage to create what we're going to call genetic maps, and we'll cover that in our next video.